Hi everyone, my name's Jeremy. I'm a postdoc in Nick Lohman's group here at the University of Birmingham. And my work involves working both here and also abroad in Sao Paulo in Brazil with our collaborators. And what we tend to look at are arboviruses and how we can basically better track them. And most of the methods I tend to use tend to be sort of looking at using metagenomics and sort of long read approaches. Previously, before I joined our group, the group sort of focused on things like any sort of outbreaks that happened and provided sort of both genomic and bioinformatics support. This includes when Ebola happened. Previously, I was coincidentally in Brazil at the time when the Zika pandemic happened. And then obviously, I think Rob mentioned, and I'm sure a number of people's research has been sort of quite impacted by coronavirus. Thankfully, I'm not going to be talking about that today. But I was one of those idiots who decided, you know what, COVID sounds like something I should do or something I should help with. So while everyone else was being impacted with it, I thought I'm going to start trying to join the effort to help with the sequencing for this. So myself, Sam and Nicola joined Nick's group around sort of a couple of years ago now. And since then, I've moved on to helping with sequencing for the more recent Mpox outbreak, as well as working with the CADE consortium. And this consortium was founded and centered around the Zika outbreak that happened in around 2015, 2016 in Brazil. I'm sure a number of you have seen the photos of, say, the kids getting things like microencephaly, a lot of the other sort of more quite visceral symptoms that they're not very nice at all. But a lot of the work that was sort of centered around that project has led to what I'm working on currently. And the general way we go about it with any of these sort of outbreaks or any of these pathogens is using a set of tools to develop an amplicon scheme. What that means is we basically have a set of two different primer pools that cover the entire length of the genome for that particular virus. And from that, we can amplify and generate a full consensus sequence with a great depth of coverage. And while this approach works quite well for a lot of cases, for all of these outbreaks, for example, there are limitations, such as, for example, if you don't know what you're actually sequencing. And with a lot of arboviruses, one of the problems you have is that they tend to have very non-specific symptoms. You'll have people having sort of acute febrile illnesses. It can affect people in very different ways. So having different approaches, such as metagenomics, means that you can target these in different ways. And with nanopore sequencing, a previous method worked on by both Dr. Quick and Dr. Ingrid Claro, who's now moved back to the University in Imperial College in London, but was previously working in Brazil on a PhD at the time. They were able to develop a rapid viral metagenomic assay. And for this, the main thing you need to just understand is that it just allows you to have a non-targeted, non-reference-based approach that allows for long-read sequencing of the entire genome. So that way you can basically have the entire genome, say for something like Zika, in a single read, which if you have no reference, if you have no idea what you're looking at, and what you have is potentially novel, such as cases as the ones I've got on the screen now, it can mean that we have the potential ability to identify new arboviruses. Now, when I use the term arbovirus, that basically refers to any virus that is insect-borne in origin, but for the case of my work, I'll mainly refer to mosquitoes. And one of the major issues we have with these is that we have no real way of detecting them until they reach this sort of zone of emergence. So usually the only time you really see this is because of the creep of urbanization, of global warming, of climate change, or sort of just general deforestation. And all of these, I'm sorry, this isn't very positive or happy, is it? But unfortunately, with a lot of things more recently, now perhaps with the current government compared to sort of what it was under Bolsonaro, you are seeing some decreases in deforestation, but you still have a large amount of human impact, and that encroaches into these sort of wild spaces or times where the viruses are in a sort of sylvatic cycle. So the vectors, the animals that they will feed on, tend to be sort of wild animals that are not really that affected by the virus. They don't experience massive amounts of symptoms. They're not going to try and, because ultimately the virus doesn't really want to kill its host. But when you get these spillover events, you do end up with outbreaks such as Zika, such as dengue, which you tend to get every year, other ones including things like chikungunya and West Nile virus and yellow fever as well. But in addition to those, you are starting to get some novel viruses as well. So the aim of my project was basically to develop a proof of concept assay that can allow us to identify any viruses that are present, as well as having some method of identifying the host, mosquito species, and any vectors that were basically, any animals that were being fed on. And a lot of that work has sort of been involved in a recent workshop that I was involved in, and a couple of the others from our lab were involved in, where we got to go to Brazil and have slightly nicer weather than we have here at the moment. Although incidentally, I have got a couple of colleagues from Brazil who have decided to come over to England at this time, and they've gone from a lovely sunny Sao Paulo to basically grey skies and nice and lovely English weather. But one of the things we were able to do there is do a bit of field work at this place called Horto Florestal. And over there, we were able to see how the mosquitoes are collected. And that's normally done by mechanical aspiration. So you will basically, or some of the people who collect them will almost let the mosquitoes land on them, start to feed, and then just quickly suck them up with a little vacuum thing. Now, having seen some of the viruses in them, I'm not sure I would be quite as comfortable as those people were. But it is very effective. You can also use things like CO2 and a number of other methods to collect mosquitoes. For most of the work I did, we worked with a group at a place called Susain, which work a lot with the Sao Paulo Zoo. And over there, they were able to collect them just by having these very large white sheets. And basically, as the mosquitoes will feed on animals nearby, they will just sort of land in these areas. And you can just very quickly and simply just suck them up with the aspirators. So from this kind of thing, we were able to have, so that was basically how we were able to collect them. And then through that, I took the previously existing protocols that we had for random metagenomics and adapted it for this case. So for what I needed to do for the project I had, I wanted to only ensure that any viruses that I was finding were going to be actively infecting the mosquitoes. So for that, we basically just lop the heads off and only sequence that part. That way you can be a lot more confident than if you were just sequencing parts of the abdomen. It's possible that you'll find more viruses, but you can't guarantee that that is actively infecting the mosquito and can potentially transfer to other hosts. The technique we use, as I mentioned vaguely before, is called Smart9N. I'm happy to go into this in more detail if anyone has any questions about this later. But the more important thing for you that you need to know is just it involves taking the viral RNA, generating complementary cDNA with the Smart9N oligo. And what that allows you to do is, during the amplification, introduce the indexing as well. So you can basically have a barcoding reaction where you can multiplex your samples at the same time as you are amplifying them. There's a bit more to it than that, but for today, I won't go into it in too much detail. And that works really well for viruses because they're quite small genomes, generally speaking. I mean, even large ones like Mpox will be, 200,000 to 300,000 base pairs at the maximum. But for most of the ones that I work with, they tend to be sort of 10 to 20,000 bases. So we're able to get the entire virus at times in single reads. But for the vertebrates that we'll be working with, for a lot of the mosquitoes we'll be working with, obviously, while it would be nice to be able to sequence the entire thing, we're normally unable to. So for that, I use a thing called DNA barcoding. And for this, there are a lot of different barcodes that you could use, or different sections of DNA that are highly conserved that you could use. In my case, I found it was the easiest to use the cytochrome oxidase C in the mitochondria, just because that has a very well-established database for it. And there's also been a lot of other people who've tried working on this before. So for that, they had, I took a number of different primer schemes that had been tested before, mostly done with Sanger sequencing, but in all honesty, it's quite easy to just test and adapt them to see which ones would be suitable for nanopore sequencing or for running with different types of enzymes. And from this, I was able to find a set of 4 different primers that I was able to just amplify directly from by taking the blood-engorged abdomens, extracting the DNA from them, and then just amplifying. And the way I set up the reaction was such that it was possible to combine this with the arbovirus metagenomics assay. So you're able to have an amplicon-based approach combined with a metagenomic approach using nanopore sequencing and using the same sequencing chemistry. And generally speaking, this is sort of a vague sort of display, just sort of showing more or less how the sort of workflow in the lab works. Following on from this, we were able to run about, say, 10 or so mosquitoes per library and still get enough coverage to see all the viruses and see what the animals were feeding on. So generally speaking, for the bioinformatics side of things, the first tool we use for any kind of viral identification for metagenomics is something called Kraken-2. And that is a relatively simple database. In my case, I was just using the one for viral references. And from that, you're able to sort of have a very simple display in a sort of table format about what kind of pathogens you might have there. But you can't guarantee that what you're seeing here actually corresponds to actual reads. So for that, I had a very simple sort of pipeline that I had just developed, which took a bunch of very simple sequence alignment tools to generate a consensus sequence based on the potential GenBank references I was able to extract from the Kraken-2 report. And from that, when I actually look at those files in more detail, so such as this one, which is, for example, this is from the Namdin virus, where it had about 9,500 reads total. We're still able to get a decent enough level of coverage, at least for a metagenomic assay, and see that what we were actually getting were genuine reads. And then for the blood meals and for the mosquitoes, we just used a form of Minimap2 with a particular wrapper that Sam Wilkinson designed. And what that allows for is it gives you some degree of idea of quality when it comes to mapping the entire reference database from barcode of life. And when you're comparing that, all of those sequences and references that are there to what you have, you can get a better idea of what kind of animals that they've been fed on. But there are some limitations to that. For mosquitoes especially, we were only able to find there are only 150 or so species referenced in bold for Brazil. Just for one genus such as Culex, there are over 900 species that have been identified. So obviously this can be a bit limited. So for that, we had to use a slightly different approach in the end, which involved non-reference-based polishing, where basically I used a program called Racon. And what that allowed me to do was basically take all of the FASTQ files, basically extract all of the ones that had the primer sequence for the mosquitoes, and then just polish all of those and compare them against each other. And then from that, I could generate a consensus sequence and just blast that. And so what that did was it would basically, because when I actually looked at the sequences that were being extracted, I had a very high depth of coverage, especially for the amplicons. And there were also some nice overlaps as well. So it ended up being quite easy to have. And from that, we had the sort of proof of concept of the assay where we were able to identify all three components. But just having that alone, it's much easier if you can actually visualize what you have with the data and how you can basically use that. So this is using this is a type of graph called the Sankey plot. And what it allows for is the sort of nice idea of being able to see visually sort of which animals are being fed on by which mosquitoes and how those kind of links can potentially go. And then if we add the actual viruses to that as well, what you can see is something like the Chilean flamingo is being fed on by multiple species of this genus Culex. And of those, there are multiple viruses that are already infecting those mosquitoes as well. If we also look, we can see that some of the species, such as Declarata, also feeds on humans. So you have that potential concept of potentially certain flaviviruses, certain other viruses, being able to transfer from one host to another. And with that, basically, that's more or less what I have so far. The aim now is to basically have a preprint, which we're currently working on. But from this, we have now a proof of concept assay for arbovirus surveillance, using this sort of combined approach. And the aim is to sort of take this and further streamline it. So we have for example, I work with, so recently NEB has released a new enzyme called Q5 blood. And what that allows for is potentially you can do direct PCR amplification directly from blood without any form of extraction. So currently I'm trying to develop an assay testing that to see if that can be used. And then in theory, you can take this entire assay and basically make it entirely based in the field without the need of a lab, using something called a bento lab. I don't have that slide on here, sorry, but that doesn't matter. Anyway, and just as a final thing, I'd like to thank everyone involved in the project. Especially like to thank the 3 PIs involved in CADE. So that would be Nick, Nuno, and Esther, especially Nick for being my PI these past 2 years and for dealing with me for most of this case. I'd also like to thank everyone in my lab, as well as the guys in Brazil, especially Professor Karen and Dr. Lilian Guimarães, who provided the samples. And just as a final thing, I'd like to thank everyone involved in the CADE workshop as well. Thank you very much.